A single stain tends to be enough to show the fundamental morphological characteristics of the majority of microbial cells, such as their relative size, shape, and characteristics when grouped together. This type of staining is known as simple staining. Several distinct microstructures can be seen thanks to the chromophores in these stains, which react chemically with particular cell components or structures. Differential staining techniques have been developed to gather more information. Differential stains, so named because of their distinct reactions to various cell types, are crucial for classifying organisms. The Gram stain is the most significant and frequently used differential stain for identifying the composition and structure of the cell walls that account for the two groups' varied reactions to the Gram stain. The acid-fast stain distinguishes between a significant class of bacteria called Mycobacterium based on the lipid content of the cell, in a process that is very similar to that of the Gram stain bacteria. Bacteria can be classified in general as either Gram-positive or Gram-negative microorganisms based on how they respond to the Gram stain.
Procedures. Smears from cultures of 2 microorganisms were prepared. The smears were heat-fixed and air-dried. The slides were placed on a staining rack and each smear was flooded with crystal violet. After staining for 1 minute, the crystal violet was washed off with tap water. Excess water was drained. Each smear was flooded with iodine. After 1 minute, the iodine was washed with tap water. Bibulous paper was used to remove the excess water by lightly blotting it with the excess water. Each slide was tilted and decolorized with 95% ethanol until the alcohol draining from the slides became colorless. The slides were washed briefly with tap water and the excess was drained off. For about 20 to 30 seconds, the smears were counterstained with safranine. Then, the slides were washed briefly with tap water and blot dried. The slides were examined under the oil immersion objective.
Hans Christian Gram developed the Gram staining technique in 1884 to differentiate bacteria based on their cell wall structure. Gram-positive bacteria have thicker peptidoglycan layers and hence appear purple, whereas Gram-negative bacteria have thinner peptidoglycan and outer membranes and thus appear pink/red. Because of its capacity to classify bacteria into discrete groups based on cell wall composition, this differential staining approach became a cornerstone in microbiology for bacterial classification and identification.